Hello my geniuses, welcome to another biochemistry lecture series. It's your great science teacher Lorenzo, and today we'll be breaking down and dissecting purine metabolism. So in our last video we did pyrimidine metabolism, so this one will be doing purine metabolism. So if it's your first time in my channel, make sure to subscribe and turn on the post notification button so that anytime I drop any video you'll be sure to be notified. And if you want me to make any video for you, or you are studying and you seem to not understand some topics, just put it down in the comment section or just send me a message, and I promise you that I'll make that video for you. So let us jump right into the video. Here in the introduction, what is purine metabolism? First of all, we have to know what purine is before we now know how to form purine. So purine metabolism, in purine is a nucleotide just like pyrimidine. That is what you know, and both of them are important information of nucleic acid. So when you polymerize nucleotides, you get nucleic acid. Just an example. So you have to couple two or more nucleotides together to get nucleic acid. Say for instance, A is complementary to what? C, which is adenine, is complementing to thymine, while guanine is complementing to cytosine. That ATGC. And I told you that when I was giving what is called it, I gave you a pyrimidine basis, but they are just called CUT, cytosine, uracil, and thymine. But here is just AG, which is adenine and what? Guanine. Don't forget. So when you form these people, you utilize them for nucleic acid synthesis. So what product are we expecting here? We are expecting what? AMP, which is adenosine monophosphate. We're expecting guanosine monophosphate, and we are expecting what? Inosine monophosphate, IMP. So we are also there are also minor purine bases. So there are also minor purine bases. Same we talked about in pyrimidine. Purine, the structures of adenine is what? 6-aminopurine. Don't forget, adenine is what? 6-aminopurine, while guanine is what? 2-amino 6-oxo what? Purine. So both of them are important structure, just like the one I gave you for pyrimidine molecule. So now in this one, purine can undergo too. So naturally we cannot eat. There's no food that is rich in what? Purine or pyrimidine. No, our body synthesize them, and we can also get them through salvage what? Pathway. So don't forget. So in this one we are doing de novo synthesis of purine biosynthesis. That last one we have done de novo synthesis of pyrimidine. So we are doing de novo synthesis, and here too we also do the salvage pathway too of purine synthesis. So one thing you want to have to understand, the first reaction before you start purine synthesis, you have to know that we talked about the structure of pyrimidine. The pyrimidine is a heterocyclic ring, which is just one. It contains a 1-pyrimidine ring, which is just like a benzene ring. It's a 6-membered ring. But for purine now, you have 2 rings combined together. You have a pyrimidine ring and an imidazole ring. So we have 2 rings in purine and 1 ring in pyrimidine. 2 rings in purine, 1 ring in pyrimidine. The two rings in purine is one is pyrimidine, while the second one is imidazole. While the only 1 ring we have in what? Pyrimidine is just one 6-membered pyrimidine ring. But in purine we have a 6-membered ring and a 5-membered ring fused together. So don't forget it. So now in the 6-membered ring, what are the components? What are we utilizing here? We tell you the difference between purine and pyrimidine. Here that in purine, first of all you have to form the sugar. On the sugar you now have to form the ring on the sugar. You have to put various components on the ring of the sugar to form everything you want to look for. But in pyrimidine you have to form the ring first before you now attach the sugar. You know the difference. Pyrimidine you form the ring, attach the sugar, while here you on the sugar you put the ring. You form the ring on the sugar. Okay, now you form the ring so there must be a sugar before you can form the ring. But in pyrimidine you have to have the ring first before you now attach the sugar. That is the difference. Yeah, on the sugar molecules now, what are we utilizing for purine metabolism? So we are utilizing some components here. So first of all, we are utilizing CO2. CO2 is usually synthesized from the 6 carbon in the pyrimidine ring. The 6 carbon in the pyrimidine ring. So we are getting the CO2 from bicarbonate there. So when we attach the CO2, information of all this, because the first product of purine is IMP. So we are forming IMP first. So it takes 10 steps to form IMP, and these 10 steps are very, very important, that you must add all these things to the sugar to get pyrimidine. So the first thing here, we are adding CO2. We add CO2 derived from carbon 6. The next one is what amine group. The amine group we're attaching here is aspartic acid aspartate to carbon 1. That is on the for the nitrogen carbon we are adding here is in carbon 1. The next nitrogen carbon is on carbon 3 and carbon 9, which is the amide group, which is glutamine. Don't forget glutamine, carbon 3 and carbon 9. That is the nitrogen we are adding to the reaction. So we are talking about the CO2, we are talking about the nitrogen in carbon 1, and which is the amine group, and the amide group in carbon 3 and carbon 9. The next one is glycine. Glycine is added to carbon 4, 5, and 7. Glycine, carbon 4, 5, and 7. Don't forget. The next one is formyl group now. The formyl group, which is N raised to power 10 formyl what trihydrofolate, added to carbon 2 and carbon what 8. Don't forget it. So all these things are important for you to form a good purine. So the first product is IMP. From IMP, you can form what AMP, which is adenosine monophosphate and guanosine monophosphate. So let us start the reaction now. So the first thing we'll be doing in the biosynthesis of purine nucleotides, what are we going to do first? First of all, I told you that you have to form the sugar, then you build the ring on the sugar. So first of all, in making the sugar, I told you that in the oxidative phase of HMP shunt, what happens? The oxidative phase of HMP shunt, you are forming what? Ribose 5-phosphate. So for formation of DNA and RNA and formation of nucleotide, you have to do what? First of all, the ribose 5-phosphate is converted to PRPP, that is phosphoribosylpyrophosphate. Pyrophosphate. So catalyzed by what enzyme? Phosphoribosylpyrophosphate pyrophosphate kinase, or PRPP synthetase, utilizing ATP. So when we utilize ATP, breaking it down to ADP, using the enzyme PRPP kinase, that is phosphoribosylpyrophosphate pyrophosphate kinase, to convert what? Ribose 5-phosphate to what? PRPP, phosphoribosylpyrophosphate pyrophosphate. So when we form the sugar, we cannot form the reaction. I told you that information of, first of all, you have to form IMP, which is inosine monophosphate or inosine monophosphate. So how can you start? In the first reaction, first of all, you utilize your your number 3 and number 9. In the number 3 and number 9, you are donating what? Glutamine. So the glutamine there, the amide group, what happens? You synthesize what we call phosphoribosylamide. Phosphoribosylamide, we call it PRA for short. Phosphoribosylamide. So when you transfer glutamine, which is the nitrogen in number 3 and number 10, when you are transferring the nitrogen number 3 and 10 to carbon 1 of PRPP, so when you transfer it to carbon 1 of PRP, what happens? You will be forming what? Phosphoribosylamide, catalyzed by what enzyme? GPAT. So you remember GPAT, glutamate phosphoribosylpyrophosphate pyrophosphate amidotransferase. So when you transfer glutamate in carbon 3 and carbon 9, what happens? We transfer it to carbon 3 and carbon 9, you'll be forming what? Phosphoribosylamine, catalyzed by GPAT, glutamate phosphoribosylpyrophosphate pyrophosphate amidotransferase. You transfer the glutamate to carbon 1. Don't forget. Phosphoribosylamine, what happens to it? It is now added with glycine. So glycine is added. So glycine provides number carbon 4, carbon 5, and carbon 7. So when glycine is added to phosphoribosylamine, what happens? It will form what we call 5-phosphoribosyl glycine amide. 5-phosphoribosyl glycine amide. Don't forget it. Or you can call it glycine amide ribonucleotide. Glycine amide ribonucleotide, or you can call it 5-phosphoribosyl glycine amide. So when you are adding glycine to PRA, which is phosphoribosylamide, you are forming what? 5-phosphoribosyl glycine amide, or glycine amide what? Ribonucleotide. So what enzyme is catalyzing this reaction? It's a synthetase reaction. Why? Because you are using ATP. You can either use the enzyme 5-phosphoribosyl glycine amide synthetase, or you use glycine amide kinosynthase. So this enzyme, they are the same thing. So when you have a synthetase in the reaction, so you are utilizing ATP, and this provides carbon 4, carbon 5, and carbon 7. So the next one, now we are using our formyl group. Our formyl group, we are adding to carbon 8. Our formyl group, we are adding to carbon 8. So how are we adding? So we are using formyl transferase to transfer formyl group into what? 5-phosphoribosyl glycine amide. So when we transfer formyl group into 5-phosphoribosyl glycine amide, what will it form? It will form 5-phosphoribosyl what? For N-formyl glycine amide. 5-phosphoribosyl N-formyl glycine amide. So using what? Formyl what? Transferase. So we are provide carbon 8 now of this reaction. So after forming five phosphoribose N forming glyceramide, the next reaction, what are we going to do here? We are going to now, because we are provided carbon 9 to form PRA. So the first one after we form PRPP, that will transfer amine group, the amine group to what PRPP at carbon 1. We form what phosphoribosamide for carbon 9. Now we want to form carbon 3 now, the carbon 3 part. So we have formed carbon 4, 5 and 6 using glycine, and we have formed carbon 9. So the next, and we have formed carbon 8. So the next one we are forming is carbon 3. Now we would also transfer glutamine now. So glutamine will donate what? The amino group. So glutamine will donate the amino group. So if it donates the amino group to 5-phosphoribose N forming glyceramide, what will it form now? It will form what? 5-phosphoribose N forming glycine amidine group. So we are forming glyceramide. So glutamine will be converted to glutamate, dropping off the ammonia. Then what will happen? We are utilizing ATP. So what will reaction with which enzyme will it be? Synthesis. So we'll call the reaction 5-phosphoribose N from a glycine amidine synthetase. So anytime ATP is being utilized, we are using a synthetase what reaction. Don't forget. This reaction, we are going to close the 5-membered ring. We have formed, this is the last part for the formation of the 5-membered ring, which is the imidazole ring. So we are good. This reaction will be catalyzed by what? 5-phosphoribose amino imidazole synthetase. So ATP will be utilized to close the ring. The last one we formed was 5-phosphoribose N forming what? Glycine amidine. When we give the carbon 3, we've put the carbon 3 of the 6-membered ring. So we that's how we form it now. For this one, I want to close the 5-membered ring. So if we close the 5-membered ring now using this enzyme, so we'll be converting 5-phosphoRibose N forming glycine amidine to what? 5-phosphoRibose amino imidazole, catalyzed by a synthetase reaction utilizing ATP. So now we have closed the 5 ring. Now let us go to the 6 ring. Now information of the 6 ring, what happens? 3 of the 6 rings are already present in this reaction, which is 4, 5 and 9. So 4, 5 and 3, 4, 5 and 3 has already been formed. That is 3 of the 6, 4, 5 and 3, that is 3, 4, 5 have already been formed. 4, 5 in glycine and 3 for what? Glutamine. So it has already been formed. So we're leaving us with CO2 in carbon 6, carbon 1 aspartate, and carbon 2, which is what? N-formate trihydrofolate. So these 3 we are going to source it and add it to them. So for the 5-membered ring, we have formed them already, and also leaving also part of what we have formed, we now use 4, 5, which is glycine, and 3, which is glutamine. So now information of the we have closed the ring already. Then in this next reaction, we have formed 5-phosphoRibose amino imidazole already. So which makes us close the 5-membered ring. No, 9, the 6-membered ring. The next one, first of all, for carbon 6, we will now bicarbonate to donate CO2. So when we carboxylate the reaction, what are we going to form? If we carboxylate the reaction utilizing about ATP, what are we going to form? We'll form what? 5-phosphoRibose carboxy amino imidazole. That's what we form, catalyzed by a synthase reaction. So 5-phosphoRibose carboxy amino imidazole synthetase using an ATP. So ATP will be utilized in all this reaction. So immediately we are donating CO2 for carbon 6. So now we have added CO2 to form the carbon 6 part, which is the 6th carbon of the 6th ring of the 6-membered ring. So the next thing we'll do here now, we'll now add our aspartate. So we'll be adding aspartate into the reaction. So adding aspartate to this reaction, what happens? Adding aspartate, will be forming what? We'll add aspartate to 5-phosphoRibose carboxy amino imidazole. So what are we going to form? We are going to form what? 5-phosphoRibose succinyl carboxy amino imidazole. Don't forget 5-phosphoRibose succinyl carboxy amino imidazole, catalyzed by also a synthetase reaction. Catalyzed by what? A synthesis reaction. So now when we are transaminating amino aspartate, so if we put aspartate, we are getting succinate. So if we transaminate aspartate, we get succinate, and this succinate is imputed into the reaction. So since we are also using this, so we have converted from 5-phosphoRibose carboxy amino imidazole to what? 5-phosphoRibose what? Succinyl carboxy amino imidazole. So using a synthesis reaction. So the next reaction now, the 5-phosphoribosylate succinyl carboxy aminoimidazole fumarate will now leave the reaction now. Fumarate will leave the reaction, catalyzed by the enzyme adeno what succinate lyase, adeno what succinate lyase, adenylo succinate lyase. So if we use adenylo succinate lyase, we remove fumarate from the reaction. So what are we going to form if we remove it from the reaction? We'll be forming what? 5-phosphoribosylate adenylo carboxy aminoimidazole. 5-phosphoribosylate adenylo carboxy aminoimidazole. So fumarate will leave the reaction. So if fumarate will leave the reaction, remember we are using a lyase reaction to remove fumarate. So what is the next thing now? After we have formed this one now, so which means we have added to carbon 1, we have added to carbon 6. So the next reaction now, so you remain the formyl group now. So the last one, formyl will be added to what? 5-phosphoribosylate adenylo carboxyl aminoimidazole group. So it will be added to the group now, which is the last one. So we'll use formyl transfer to transfer formyl group into this reaction now. So if we use formyl, we not transfer formyl group, what are we going to get? We'll get what? 5-phosphoribosylate N-formyl carboxy amidoimidazole. Get 5-phosphoribosylate what? N-formyl what? Carboxyl aminoimidazole group. So when we now get it now, catalyzed by what? N-formyl transferase. So formyl transferase we transfer formyl group from what? Trihydrofolate into the reaction to form what? 5-phosphoribosylate N-formyl what? Carboxy aminoimidazole. So when we form it now, the last reaction, so we have formed it, there will now form IMP. So this reaction now, the last one we say, which is 5-phosphoribosylate N-formyl carboxyl aminoimidazole, is converted to IMP by what? IMP cyclohydrolase. So we'll dehydrate the reaction. So if we remove water, we'll close the 6-member ring. After forming IMP, which is the parent compound for purine synthesis, so 10 steps makes us to form IMP. We first form PRPP, then we take 10 steps to form IMP. So after we have formed the IMP, what happens? So IMP is catalyzed by the enzyme adenosine synthesis utilizing GTP. So when we convert GTP to what? GDP, we condense IMP with aspartate. IMP with aspartate, removing water. So when we condense IMP with aspartate, removing water, utilizing GTP to GDP, water will form adenosine. So IMP to adenosine catalyzed by what? Adenosine synthesis utilizing GTP, adding aspartate to remove water. So the next thing we now do now, adenosine using adenosine lyase forms AMP. Don't forget, adenosine using adenosine lyase forms what? AMP. So AMP is the what? The first RNA we have from here. AMP. From AMP now you can also do the same thing. You can use the ribonucleotide reductase to form what? DAMP. You get it now? So AMP, which is RNA, you can form DAMP by ribonucleotide reductase, and you can use nucleoside what? Monophosphate kinase to form ADP. From ADP to ATP. It's very simple. So the next reaction here, the same IMPs too can use what? IMP dehydrogenase, utilizing NAD plus to NADH. So when we reduce NAD plus to NADH using IMP dehydrogenase, what are we forming? We are forming xanthosine monophosphate XMP. So from xanthosine monophosphate, so IMP can be converted to xanthosine monophosphate by using a dehydrogenase reaction. So with dehydrogenase, we are converting what? We are reducing NAD plus to NADH. So now we have formed xanthosine monophosphate. So xanthosine monophosphate can give us GMP, which is guanosine monophosphate, by using what? Guanosine monophosphate what? Synthesis. So when we use GMP synthesis, we are dropping off glutamine is converting to glutamate, dropping off amino group. Then we are also utilizing water in this reaction and ATP. So when we utilize water and ATP, xanthosine monophosphate is converted to GMP. The same thing. GMP can now be converted to GMP is a RNA, so it can convert to DNA by ribonucleotide reductase. So it's very, very easy. So we have formed IMP from the 10th step. IMP forms GMP and what? AMP. From AMP you can form ATP, which is the energy component in cell. That is ATP I'm talking about. So from the GMP you can form GTP. GTP2 is also used as what? The energy component in various cells, especially in formation of energy, especially the substrate level phosphorylation in TCA cycle, where GTP is converted to GDP, ADP is converted to ATP. So GTP is used there, and both of them, so they are used as second messenger. Cyclic AMP, they also use cyclic GMP, cyclic AMP as used as second messenger too. So it's very, very important. So we have talked about the purine biosynthesis. So that is the end for purine biosynthesis. So we have formed purine using the 10 step to form IMP, which is from PRPP to PRA, which is phosphoribosylpyrophosphate pyrophosphate amine. So we formed every steps down to IMP. Then from IMP we formed AMP and GMP. So I hope you understand everything we have discussed in this video. So we'll be moving to the purine catabolism and the salvage pathway. Stay toward the end of the purine metabolism video and you understand how DATP is formed and DGMP is formed and how they are combined for DNA synthesis.